PCR-free library preparation kit. However, the high library concentration, and more importantly, selective amplification of fragments ligand with correctly oriented adapters allow for fluorometric quantification. 3. Nexteria DNA library prep kit, ideal for large, complex genomes, for example, the human genome, and provides a shorter sample preparation time relative to the TrueSeq PCR-free and Nano library prep kits. The protocol is similar to the TrueSeq Nano DNA library prep kit.
DNA fragments. Through reduced cycle amplification, additional motifs are introduced, such as the sequencing binding site, indices, and regions complementary to the flow cell oligos. Clustering is a process where each fragment molecule is isothermally amplified. The flow cell is a glass slide with lanes. Each lane is a channel coated with a lawn, composed of 2 types of oligos. Hybridization is enabled by the first of the 2 types of oligos on the surface. This oligo is complementary to the adapter region on one of the fragment strands. A polymerase creates a complement of the hybridized fragment. The double-stranded molecule is denatured, and the original template is washed away. The strands are clonally amplified through bridge amplification. In this process, the strand folds over and the double-stranded bridge. This bridge is denatured, resulting in 2 single-stranded copies of the molecule that are tethered to the flow cell. The process is then repeated over and over and occurs simultaneously for millions of clusters, resulting in clonal amplification of all the fragments. After bridge amplification, the reverse strands are cleaved and washed off, leaving only the forward strands. The 3' ends are blocked to prevent unwanted priming. Sequencing begins with the extension of the first sequencing primer to produce the first read. With each cycle, fluorescently tagged nucleotides compete for addition to the growing chain. Only one is incorporated based on the sequence of the template. After the addition of it by a light source, and a characteristic fluorescent signal is emitted. This proprietary process is called sequencing by synthesis. The number of cycles determines the length of the read. The emission wavelength, along with the signal intensity, determines the base call. For a given cluster, all identical strands are read simultaneously. Hundreds of millions of clusters are sequenced in a massively parallel process. This image represents a small fraction of the flow cell. After the completion of the first read, the read product is washed away. In this step, the index 1 read primer is
called bases. Altogether, our DNA contains about 6 billion such paired chemical bases. There are 4 bases in the DNA alphabet: A, T, C, and G. In DNA, base A joins only with base T, and base G joins only with base C. To read the sequence of bases in DNA, samples are inserted into a sequencing instrument, where high-frequency sound waves break the DNA into smaller pieces that are only about 600 bases long. Special tags are added to the ends of the fragmented DNA. These tagged strands of DNA can then attach to a glass slide. In a sequencer, each piece of DNA is copied hundreds of thousands of times, which creates clusters of identical DNA fragments. Next, the sequencer reads the DNA, one base at a time, using different colored tags for each DNA. Check the different colored tags. This sequence of colors reveals the DNA sequence of each fragment. Powerful computers piece together these individual DNA fragments and reveal the sequence of your DNA. Then, a team of medical experts uses specialized software to analyze and compare the DNA sequences to identify the handful of variants that may be important for your medical care. Discuss the results of the sequencing of your DNA with your doctor and a genetic counselor.
The world. You may also find out some information about your risk of developing common diseases which are caused by multiple genetic, environmental, and lifestyle factors. For example, you may have DNA variants that mean you have an increased risk of a disease like type 2 diabetes. Remember though, that type 2 diabetes is also influenced by what you eat and how much you exercise. There is also the possibility of getting personal genetic information about your risk of developing a psychiatric disorder, like schizophrenia or Alzheimer's disease. Whole genome sequencing can also provide information about how you might respond to medications. Variants in your genome can influence whether or not a specific medication will work for you, as well as whether or not you might experience unpleasant side effects.
I'm Vidushi,
And I'm Mehuli.
and we're from Jyotirniwas College, and we're doing our bachelor's of science in biotechnology. And we've come here to attend a 2 days hands-on workshop in next generation sequencing. So in this, we were basically given a small, brief introduction about the wet lab part of NGS and a detailed and hands-on experience in data analysis, that is the dry lab part of NGS. We learned 3 things, that is RNA-seq, DNA-seq, and metagenomics. So Mehuli will tell you a little bit about what we learned in
In
RNA-seq.
RNA-seq, we try to determine the differential gene expressions in 2 kinds of cells, the normal one and the mutated one. This will be helpful especially in cancer detection, and today NGS is a much better technology than Sanger's. It can be used in clinical applications as well in hospitals to help in the treatment of cancers. We did in the bioinformatics part of it, we did RNA-seq by counting reads and then plotting the maps and through that analyzing it using various software like DGUST, and then analyzing it and trying to find out which genes are mutated by what extent.
In DNA-seq, we learned about different variants that might be present in the sample that is given to us by aligning it to a reference genome and basically finding out,
Looking for mutations.
yeah. And in metagenomics, we found out the various kinds of bacteria present in 2 soil samples that were given to us. So yeah, that was
It was a great experience. Yeah, and we learned a lot of things which will be very beneficial to us in future.
I'm Professor Dipshika Bora from the Department of Life Sciences, Dibrugarh University, Assam. I basically belong to zoology with specialization in entomology. I have been involved in gene expression with respect to defense response of plants, and also so from that aspect, whatever I have learned on the first day of the training, 2 days training program, that is RNA-seq, how differential expression of genes can be studied, that has been a great experience. And of course I knew a lot about this presentation of these expressions, but how it can be done, that I have come to know from this training. And as far as DNA-seq, how the SNPs, yes, I do work on diversity of silkworms of Assam from different regions and compare it with the genomes of other silkworms. And from that point of view, I was interested in SNP, like how we can compare the gene sequences between different types of silkworms. So again, it's of use for me. And the third thing, that metagenomics, and again I'm onto that field because I'm working on different, bioindicators of soil sample of different systems of tea. So that way, how we can, compare what is happening in a conventional tea system, what is happening in a where organic farming is done, so that I can compare with the knowledge that I have gathered here. So I think it has been a great experience, and it will be of use for my research and the students. Thank you.